Okay, recap from last week. So bioremediation is basically just the process of using cells and microbes to fix our environment. Typically, this is done via redox reaction, so the exchange of electrons, try to get it to a lower energy level, or they use catalysts to maybe even put it to a higher energy level, depending on if it's a oxidation or reduction. These bioremediation processes are split into three-ish categories. The first one's aerobic, of course, which means that the microbes use oxygen as the electron acceptor. Then there's anaerobic, which is just a variety of different molecules that are catalysts that they can use in order to reduce or oxidize, but it's predominantly reduction in this regard. And then finally, heavy metal, which is typically reducing that metal or attaching to the metal in order to decrease its mobility within its surrounding environment. Last week, I also mentioned a few projects from the iGEM registry and old iGEM teams, one of which was the PET microplastics. Yale did that in 2018. They used microbes to degrade PET plastics within the water supplies. These PET microplastics are very, very harmful to the environment due to their ability to, one, clog up animals, their digestive systems, and two, they have a bunch of chemicals like xenoestrogens that mess up human hormonal levels and digestive systems. Also, algal blooms. Cornell did that in 2019. Algal blooms happen when there's tons of nitrogen or sulfur that gets shot into the local waterways. In turn, these algae start blooming and growing like crazy. And then once they reach a certain threshold, they start blooming tons and they reduce these myotoxins, I think that's what they're called, into the waterways, which are harmful to the local aquatic life. Georgia State in 2019 did a coral reef algae thing. The algae was dying in coral reefs due to changing conditions within the oceans. And they were trying to bioremediate the actual coral reef algae to make them more sturdy to fight against these rising temperatures and also rising concentrations of salt. Finally, methane reduction, Michigan State 2010 did that. That's mainly just, it's like a different type of project because it's mainly focusing on gas, which is harder to work with. Okay, so now we're going to hit the equipment side of things. The first thing I wanted to cover is autoclave. It's probably the most dangerous. I wouldn't say the most complicated, but one of the most dangerous things that we work with, simply because you can either burn yourself or pop the autoclave if you're not careful. Autoclaves, if you don't know, are sterilization chambers, and they are only useful for independent lab members. So if you're a dependent lab member, then you cannot use the autoclave, at least by yourself. You have to be in there with another person, either a professor or an independent lab member. In order to get certified for these, you have to go through a short training procedure, either with the lab manager, me, or through a professor. Typically, it's done by a professor, though, because the professors are the ones that give access to the room that stores the autoclave. Autoclaves, because they're sterilization chambers, they run at around 15 psi at 121 degrees Celsius. The one that we have is a little bit off in that regard. Sometimes the temperature goes a little bit too high or too low. And specifically, the pressure gauge is a little bit off, specifically when you're taking your materials out of the autoclave. It doesn't go down to 0 all the way. So if it's right around 2 or 3, just know that it's empty, like there's no pressure or very, very little pressure inside of it. And that's due to the winding of the gauge in it. It's a little bit off. Autoclaves are used for two main things, sterilizing utensils and also sterilizing/mixing culture media. I predominantly use it for culture media because when we make LB broth or plates, we have to heat up that LB broth and plates in order to mix it up and sterilize it. Specifically, if you're not using antibiotics, you need to do that because anything can grow in it otherwise. Sterilizing utensils, that's mainly just for glassware. You don't really want to put anything plastic in it, though you can. I typically don't do that. You can also sterilize Petri dishes that are already used. That's how most labs get rid of their Petri dishes. They put them in the autoclave bin, and that trash gets put into the autoclave for sterilization reasons. I included a link to a previous how-to document where I detail how to use the autoclave that we have. I made this a while back for one of my classes. I've already linked this in the past, but I believe I did not record that lecture in the past when I presented that. So I thought I'd link it here. Alternatively, you can go to page 101 in the lab notebook /manual. It details how to use the autoclave and some of the restrictions we have on it in there. Okay, so I screen capped a little bit from the instruction manual that I made. And we're starting at step 11 because the steps previously detail how to make LB broth. But there's a specific section in the manual where it tells you how to use the autoclave. So I thought I'd include that. So here you have is our mini autoclave. It's not a full-size one because one, those typically need a lot more maintenance and they're also a lot more expensive. So if you mess up, it's kind of on your hands. So yeah, you need your materials first. You need the autoclave. The autoclave is probably going to have water in it when you first find it. It just really depends if the last person emptied it out or not. You need a bin to store whatever you're cooking in the autoclave. That's because sometimes this stuff might expand and boil out of the container, and we don't want specifically agar going into the bottom of this autoclave because then it will solidify and it will clog up the outtake. So always when you put stuff in the autoclave, put it in a bin, make sure it's metal. And then notice that on top of this bottle there is aluminum foil. You do that for the same reason, well, to keep stuff from boiling out and also to protect it from air from going into the bottle. That's the main reason. You don't want to contaminate it once you pull it out. And also this is just a container of water that we have. We use this to fix or adjust the water level. So yeah, gather all these materials, you're going to need them. This is the internals of the autoclave, and this is specifically the control panel. So this is where or one of the control panels, this is where you release the water valve in order to drain the water that's already in the autoclave. Step 12 in the instruction manual is check to see if the water level is okay. Typically, you want to make sure that there's at least 1 liter of water in there. You can go a little bit over that, but do not go under it because then the autoclave will shut off halfway through its procedure and you have to start all over again. It needs enough water in order to reach proper pressure. The drainage is right here. It's going to be on drain closed, probably if no one drained it, and then you're going to switch it to drain open. Do this only, I recommend only if the water looks especially filthy. It doesn't really matter if it's a little bit bad. You can just put it in, it'll sterilize it just fine. But if there's chunks of stuff floating in it and it looks like it's been sitting there for days on end, definitely drain it. There's a bucket right underneath it. Flip open the valve, close back the valve, and refill it with water, 1 liter. Or how I do it, I don't actually measure the water out. There's a line because so much water has been poured into it. There's a calcium deposit or something. There's staining on the inside of the autoclave. And you just pour it up to that line and you should be good. After you make sure the water level's okay, you next get your bottle and your bin and you just slide it into the autoclave. Be a little bit careful when you're sliding this bottle in because specifically these bottles are kind of high. So if you have this pulled like all the way back, then the top of the bottle is going to rub on top of the lid and that might make the bottle tip over and spill. So when you're sliding this in, kind of hold onto the handle, pull it open a little bit so you can slide it in easier. This is very zoomed out, but step 14 is close the autoclave. This is important because if you don't close it properly, it's not going to reach the proper pressure. So yeah, just like I'm doing, you grab the handle, you pull in a motion like this, and it should just snap closed. There's a hook here and there's a latch down there. It just slides right into the latch. Give it a good tug down just to finalize it to make sure it's secured properly. After that, this is the main control panel. You can't see it very well here, but there are 2 gauges, one for temperature and one for pressure. And then there's a nozzle. It looks like something off a really, really old microwave. That's pretty much what it is, actually. There's also a knob that switches between instrument and slow, which is liquids. When you're making broth, you're going to be using slow, and when you're sterilizing utensils, you're using fast, which is instruments. The timer for broths can go anywhere between 35 and I would say 45 minutes. You could go longer than that, but there's not really a point. So I just typically do 35 minutes. It's going to take around 5 to 10 minutes for the thing to heat up and 5 to 10 minutes for it to depressurize once it's completed. So set an alarm for like 20 minutes more than what you set it for so you can come back right around the right time and you don't have to waste time just sitting there. This, after it's completed, you're going to want to make sure that the dial is either 0 or very, very close to 0. If it's right here, definitely do not open that thing because you'll get a face full of steam, which would be very harmful to your face. So yeah, make sure it's right around 2 to 0. You can open it at that point. Still be decently careful because steam is going to come out of it because of the pressure that was there and how the kinetic energy of the gas is pretty high still because it's very, very hot. So be careful, put on gloves when you're pulling it out. We have gloves sitting right next to the autoclave. They're always there. They're especially long. They go up to around your elbow. So put those on and very carefully pull out your sample. Make sure once again that you're careful, especially when this is so hot, because this lid, if it gets touched, if it gets flipped over, and then that almost boiling liquid falls into this container on your hands, it's going to hurt a lot. So yeah, be careful, risk of severe burns. And that's it for autoclaves. Just make sure that you're following the guides. If you have any questions, consult this little handy notebook thing that I made. I made it specifically for our lab, so it should come in handy. Next is the microcentrifuge. If you have any experience in synthetic biology, you probably have used this before. It's not that hard of a device to use. What it does, it spins down contents by rotating at incredibly fast speeds. As you can see here, there are 2 tubes put in, you could call them wells, little slots where you slide the tubes into, and this thing spins at like up to 14 to 16,000 RPM, which is magnitudes higher than the level of Gs that we experience on a daily basis. I included a picture of the control panel. This is the RPM. There is a button right here that changes the units. Typically, it's just set on minutes minus, so that's just RPM. Get that at 14, that's times 10 to 1,000, times 10 to the third, so 1,000, so 14,000 RPM. So yeah, make sure it's set to that because typically all the procedures that we're working with use RPM. And then these are the buttons that adjust what the RPM is set at. Typically, we're working with 14 to 15. Maybe if you're just trying to mix something up a little bit, you'll go down to 8. It just depends on what you're working with. Here is the timer. Same thing, adjust time there. This is pretty straightforward. The start button, the stop button, and then the lid opener. So this thing's not going to open when it's spinning because it poses a danger to you, and it could also break the device. So you have to wait till the timer is done, and you can press or click stop. Then you can press the open button, and the lid will unsecure. When we're using microcentrifuges, what we're separating are the different particles, and these particles are going to be related or different from each other in density, size, shape, and viscosity. And if any one of those particles is varied enough to cause a difference, they're going to separate into the bottom of whatever tube you have it suspended in. So for example, you have an E. coli medium, a broth culture full of E. coli cells, and also LB broth, that's the nutrient media. And you're spinning it down in order to pellet the cells. And so you spin it at 14,000 RPM for like 3 minutes maybe. And then you pull it out, and all the cells will be pelleted at the very, very bottom of the container. And so you do this in order to pour off that nutrient media so you can resuspend the cells with whatever you're doing the procedure for, which is typically mini prepping, which we'll be doing that this week. If you have any questions about using the microcentrifuge, you can go to page 103. I specifically want to mention balancing the rotor because that's very important. If you don't balance it, you'll either damage it or it'll sound very, very weird when it's spinning. If you're working with a really large microcentrifuge, I guess it'd just be a centrifuge at that point, you have to be especially careful because if you don't balance it out, because there's so much mass in it spinning at such high speeds, it can really damage or maybe even shoot out of it and hit the wall or something. So be careful, balance your centrifuges. By balance, I mean balance it by mass, or if it has the same density, balance it by volume. So if you're working with like LB broth, that's predominantly water. So just balance it out with water because the densities are pretty similar. Also, the microcentrifuge that we have has an internal lid. It's just a little latch lid that you click the button on, you pull it out. Make sure that that's secured before you turn it on. Of course, when it's finished, you take it out, set it on the countertop, pull out your samples, and then put it back and close the lid. Next is the Thermo Cycler. This is also another very, very common device that we use in synthetic biology. Some of you may have used this. It's not that complicated. However, there is a lot of programming involved with regards to setting the settings with this device. So Thermo Cyclers, they vary temperature dependent on time. They often do this in repetitions or cycles. These things are also called PCR machines because they're predominantly used for PCR. I included a link, you can't really see with this, but there is a link to the video presentation on YouTube where I went over PCRs. That's completely cut and removed from the lab meeting itself, in case you've forgotten what PCRs are. For utilizing this, you can either follow this general procedure, or for in more detailed procedure, go to page 105 in the lab manual where it details how to use it. So first, you want to make sure it's powered on. I would hate for you to put your samples in and just leave when it's not powered on. That power is going to be on the back of the device. It's just a little knob that you turn on. Next, you open up this. There's a little pull tab here. You pull it. You load your samples, which are typically going to be microcentrifuge tubes, so very, very tiny epi tubes. They fit into the little slots in there, and then you close the lid and secure it with this nozzle. So this turning knob, what it does is it lowers the heat plate inside the Thermo Cycler so it touches the tube so the tubes can get proper heating. So you want that nice and snug against those tubes. Next, because it's turned on, there's going to be a little GUI here. And you're going to select F1 for file. It's going to take you to all the programs we've made in the past. Next, scroll to iGEM. You're going to use this little up and down arrows to scroll to iGEM and select the desired program by clicking enter. After that, it's going to bring you to the stats of the program, where it's going to tell you what the program's doing and how many repetitions it's going to be doing of it. And you click F5 to run, then it's going to ask you to input your sample volumes, and you just put in the volume that you have suspended within your tubes. Depending on the procedure or the cycle that you're running, it might take anywhere between 40 minutes and an hour and a half to finish. It's dependent on what you're doing with the Thermo Cycler. Next is the Nanodrop. Some of you may have used this already. The Nanodrop is just a simple and small spectrophotometer, or photometer, that's right, spectrophotometer, which quantifies and specifically quantifies via absorbance. So it shines light through your sample, and how much light or what type of wavelength that sample absorbs determines what your concentrations are, or if you have any impurities, which is most often the case. You often use these nanodrops after mini prepping, though you can use it like after ligations or really anything. You can use them for proteins, for RNA. We typically use it for DNA, though, after mini prepping. And we do this in order to check to see if the concentrations after mini prepping are higher than the concentrations of when we pulled them from the kit plates in iGEM. For those of you doing mini projects, mini prepping this week, we will be using the Nanodrop to measure your concentrations at the end, just to see if it worked, if we need to do something different in the future. You can check page 109 in the lab manual. It goes over utilization. It's pretty simple to use. There was an old one that we had in the past that did not have a computer, so it was plugged into a computer and there was a lot more steps involved in using it. But now, because it's an all-in-one unit, you just turn it on. There's another knob in the back that you turn on. You select the program, which is typically going to be DNA. It's right here. It's touch screen, so you just click it and then open the arm. It's going to be pretty much closed because it can get damaged if it's not closed. Pipette 2 microliters of whatever blank you're going to be using. By blank, I mean whatever your DNA sample is suspended in. So say your DNA is suspended in Tae Buffer, you're going to want to use Tae Buffer as your blank. If it's suspended in water, you're going to use water. Just make sure it's the exact same suspension that you used for whatever sample you're working with. Load those 2 microliters of sample onto a little pedestal here. It's very, very tiny. You have to put your end of your pipette right in the center of the pedestal. It's not too difficult. It's easier than it looks because the surface tension of this droplet of water is so high. It typically fits right on the pedestal, no problem at all. Though if you mess up, just pull out some wipes, wipe off the pedestal. If you closed it and messed up, wipe off that pedestal too, and then reload your samples. After you put your blank onto it, you get this arm, you close it, it will auto blank. That's something new that this unit has. We used to have to blank it manually, but they learned from that, I guess, and they added an auto blank feature. So you just close the sample pedestal. It will do the blanking. That will take a few seconds, maybe 10 to 15. Then you'll open and back up the arm. You'll get your wipes. You'll wipe off to make sure it's clean. and then you'll do the exact same procedure for your sample. So you have your DNA, you pull out 2 microliters, you load it onto the pedestal, close the arm, it'll read it, and then you're pretty much done at that point. Of course, at the very end, make sure to clean it. This is a nanodrop that labs use regularly. It's in microbiome labs, so make sure it's clean for everyone else to use. On this screen, after everything's completed, there will be a graph. I didn't really go into it here. That's more of a thing we'll do in the lab itself. It will give you a few numbers. One is the concentration of DNA that it thinks your sample has. Another one is contaminants, and there's 2 different types of contaminants, but I won't get into those here. And they measure those contaminants via the wavelength. I think it's 2060, 2080 ratios that they use. There'll also be a big graph. Typically, I think you're looking for a bell curve. If it's not a bell curve or it's like goes into the negative territory, most likely your sample is either highly contaminated or there's nothing in it. Likewise, if you're, so it's going to give your DNA readings in nanograms per probably microliter. If your reading's like 2 nanograms per microliter, that means it didn't really work. Your mini prep didn't work. You're typically looking for like at least, I'd say at least 40 or 50 nanograms per microliter. Yeah, that's it for Nanodrop though. It's fun and it's a lot easier to use than most of the spectrophometers. I don't know how to pronounce that very well, that we have in the other labs. Next, second to last one is the gel electrophoresis chamber. This is used to run gel electrophoresis. We have a few of these, some of them are missing parts, but you can put together a working one if you gather together all the parts that we have. This is the chamber itself without the gel, and this is the chamber with the gel in it. As you can see, there's a little bit blue. It's the refraction of the light going through it. So you're going to load the gel into the gel casting tray. That's this thing right here. This thing can be removed, so you can cast it in the device or outside of the chamber itself. And also, after you cast it and that gel starts drying, make sure the comb's in it so that it will form holes so you can actually load your samples. If you have any questions with regards to how gel electrophoresis works, or you don't really understand what I'm saying, click that link at the bottom. That goes into gel electrophoresis, what it's used for, and how we do the protocol. After you have your gel in your chamber, you start filling it up with Tae Buffer. This is an electrolyte solution that allows electricity to run from the electrodes. And this allows the DNA or the proteins, depending on what you're doing, to move throughout the gel matrix to the positive. I always remember that from run to red. So the DNA specifically likes going to the red because of its nature. After you have your electrolyte solution fully suspended and inside your gel chamber, you're going to load your samples. Your samples are going to have an assortment of different dyes and loading things that changes their density. You load your samples into each of the little holes. I typically separate them by about a hole apiece to make sure they don't run together. Plug your electrodes in. They're going to be plugged into a power supply. I don't remember the voltage. I think it's 100 to 140 voltage that you're running through this. And then you run into a completed. That's just dependent on what the size of your DNA is. I would say this is one of the more complicated things. There's a little bit of prep you have to do while using this. And you also have to have a little bit of knowledge with regards to what you're doing in order to use this equipment. And this is probably the easiest. It's just a glorified light bulb. It's the transilluminator that we have. It's a simple UV lamp. It's used to visualize fluorescent dyes in gels or cultures. Say, for example, you just ran a gel in your gel electrophoresis chamber. It has ethidium, or at least we use ethidium bromide, which is a fluorescent dye. And once you have that dye binds to the DNA, so you take your gel out of its container, you put it on this, and then you shine a fluorescent light through it, and that in turn will make the DNA fluoresce on an image. We can take pictures of it. As for utilizing it, make sure when you're using this, always put on eye protection. You really only need eye protection for the ones that are exposed, though. With the camera one, because it has a enclosure, you don't have to worry about it. It's not going to get into your eyeball. We have face shields there in our lab and also in microbiome if you need them. After you have your sample loaded and you have your eye protection on, you can turn it on and make sure, of course, your sample's loaded. Quickly, I specify this, quickly get your measurements. When you're working with DNA will denature pretty rapidly if it's exposed to UV for periods of time outside of its comfort. With cells, if you're working with bacteria, it will just kill them outright if you leave them there for too long. So yeah, quickly get your measurements. If you're taking pictures, flip it on, keep your hand on that button, take your picture, flip it off. It could be a really, really fast procedure, less than 5 seconds. After that, because these are used by many, many people, turn the device off and clean the sample area, whether that's with, if you're using microbes, use some sort of antimicrobial wash. If not, use like ethanol, wipe it down.